I know you're busy either talking about that test or getting ready talking about that test. That's why we had some call-ups for absences. So I'm happy to say your group is really good about being here. So that's very good. I appreciate that. I wouldn't get up this early to listen to me. I don't even like me. Nevertheless, all right, we're on the way. So let me begin where we kind of left off. I mean, I've had a couple of questions. Again, the test will be a week from today. I guess just all make it. It's got to be in pencil. The machine will not work with pen. I have a handful of extra pencils. Bring them. Nothing flaws you. Not with lack of prepared. Number two pencils. Mechanical pencils. Pick a pencil. They all work. Not a pen. That will annoy me, and we don't want me annoyed. Also, I know you're going to be depressed about this. The following day, which will be the third, I will not be here. I see tears. It's okay. I have a planned absence. Another one of those planned absences. And undoubtedly, depending on where my wife and I are signal wise, most likely I will just send you something recorded. Vis-a-vis the exam, it's not that I don't want to be here and have things thrown at me post exam, though that there's method to the madness. Uh, the grades, the only thing you're to be interested in, I've said this before, the only thing on Blackboard when you see a grade is what it says final grade with scale, assuming there's a scale applied, and there usually is. That's the only thing, that letter grade. I don't want to hear a word about points ever. I don't want to hear a word about percentages ever. I don't care what they tell you in nursing or some other school. We are commissioned to give grades. If they're telling you about this, you got to get a B thing. The grade that you want to be at a minimum is called a B minus. If you're in health sciences, it's a little bit more forgiving. Just throwing it out there. Drives me crazy. I don't care if that's if they got an issue. Have the nursing department call me up. They know who. Believe me, they know. Yeah, we'll go from there. And then and down the road, if you need to look at those, I'm not going to sit here and bring them in and waste time. That's what office hours are for. You want to come in and look at the stuff, figure out what you got wrong. Maybe you don't. It's okay. That's that's what you make appointments for. I'm not going to sit here and spend a day going over tests. Don't do that. We move forward. So enough of that lecture. Sorry, I don't mean to be like that, but the reality is there's a lot to cover, and that can be covered elsewhere. This is a brief review of vesicular transport. Again, without going through all the motions, bringing it in as endocytosis. There's a distinction, though they're really the same thing, between phago and pino. One applies solids, one applies liquid or oils. And exocytosis, you see at the end of this part of the presentation, is basically the reverse, spitting it out. Spitting it out. Transcytosis is something that I don't think I will not ask you about, but you will begin to see down the road how things transit their ways through just going through the cell as a boundary and being processed elsewhere. It's kind of how that is. And the whole of idea, there's really a lot of elegant controls of moving these things around. It's not like magic. They get pulled in. You'll see I have an animation to show you toward the end. Little roadways and passageways are basically escorting these membrane packages to wherever they're going and bringing the other stuff to them. It's not random. That would be the way to describe it. So again, all these vesicles have proteins. Typically, they involve receptors more than anything else. Undoubtedly, it's selective. Okay, so basically, that substance there's going to be a receptor for it. But pick a molecule, and it will latch onto it. And again, the big problem when pathogens hijack that becomes significant. That's where we get into the microbiological aspects of things, where bacteria and viruses are able to do that. That's very interesting when we get into that in microbiology. If you're, and I think all of you have to take micro at some juncture. Try to make it a time when I'm not teaching it. There was a joke there, but everybody just kind of looked at me blankly. It's okay. Okay, and then what it's going to do, it's either going to go straight through or it's going to fuse with a lysosome. And again, that lysosome, and this is all the time, anything that enters the cell, and particularly true when we're looking at white blood cells that are scavenging around, it's going to undergo some type of processing there. It may just be to digest it, it may be to destroy it. That's really what it's all about. And we're going to go over that mechanism more than one time. perhaps in more detail in AP2 when whoever you have teaching you that. So that we call it a phagosome. That's the vessel that comes in. They move, they sort of scooch around like a baby would scooch around on the floor. And you'll see this as well. It's like the cytoplasm, it's a liquid in a gel. And so and these structures, I mean, we have this remarkable ability, these tiny filaments that can be assembled and disassembled. It's almost like I have an arm this way and I can disassemble this arm, make it into that arm. And in doing so, when you push on the membrane, the cytoplasm flows in that direction. And that's called amoeboid motion. And most students have seen an amoeba under a microscope from pond water, because that's pretty much, biology 101 in high school class. You get dirty pond water, all those neat things spinning around and all that good stuff. Rotifers, lovely. So voila. So this is what they're talking about. It's typically size molecules that couldn't get through it for whatever reason. Here are the receptors, and bang, in they go. Pinocytosis, same deal, okay, because it's fluids or oils. Same thing. Environmental sampling is something, and I touched on it before, very significant, because that's the way our immune system is keeping in tune. You're going to see that in a lot of areas. You're going to get, when you get to immunology in AP2, you're going to hear about dendritic cells. It's kind of like they have like a lot of tree branches, and you saw them in the skin with Langerhans cell. You see them in the liver with something called Kupfer, capital Kupfer cell. They're constantly sampling. Your immune system is like any other system. It's checking the environment, and it does so it keeps itself in tune with that. Again, that's more of a microbiological idea. It's something called the hygiene hypothesis, and I discuss that in micro, which is to me very, very interesting. Sometimes we're overly clean, if that makes any sense to you. And there's ample examples historically of all of this. So samples the environment. Yeah, nutrient absorption occurs in the small intestine. I'll buy that, because typically they're in the liquid phase or oil phase. And membrane is conserved. If you're pulling in membrane on one end, whatever you're spitting out, you're pushing the membrane at the other end. And indeed, maybe that will temporarily distort the cell, but it's not like the membrane is disappearing. It's always conserved, so we have a certain degree of constancy. In other words, they're bringing us, whatever they're bringing in, they're typically spitting out more or less an equal amount of stuff. And they're just showing it to it, don't care about it. Go receptor mediated. This is much more specific, okay? And typically, and I'm not going to ask you about clathrin-coated pits. I say that for my advanced physiology, but the big idea is right here. And obviously, there's other things that are part of it. These LDLs are low-density lipoproteins. They are problematic. When we look at people who have what they call cholesterol problems, or they have lipid irregularities, and somebody a relative, is taking cholesterol medicines. They're really not about cholesterol, they're about these things. And a lot of times, if it's hereditary, they're missing the receptors. And so rather than be able to bring it in the cell to be processed, it sits in the bloodstream. And if there's damage in areas of the bloodstream, what happens is that can begin to, if there's an inflammation inside a blood vessel, it'll attach to it and start to damage it. And that's where we begin to see fatty deposits. It's got a fancy name called atherosclerosis or arteriosclerosis, hardening of the arteries. And these are fatty plaques. And this is what happens as folks get older and older. And it's really one of the things that causes people to lose legs, have heart attacks, things like that. So it's a really important kind of thing. And one of the ways, typically, when you treat those things, it's about diet and exercise. Okay? But sometimes when people have these hereditarily based problems, they utilize these various agents called statins. And you'll hear that name as you go on career-wise. I have my issues with that kind of stuff. I don't know that I think they're, like a lot of things, are overprescribed, but that's me rendering an opinion, not issuing a fact. So nevertheless, these are typical things, certain kinds of enzymes, these LDLs, insulin, unfortunately, viruses, toxins. Diphtheria is nasty. When you get a tetanus shot, if you did not know, you're getting immunized for diphtheria as well. 3 things. It's what we call a trivalent immunization: diphtheria, pertussis, whooping cough, and tetanus. So that, and typically you're getting that shot not because you're afraid they're going to get it. They want to make sure that you're kept current. And then there's different kinds of things. We'll talk about caveolae. That's typically a little bit, they're different from the protein coat. Okay, there's other things that can put, here's tetanus toxin can make its way in. So this is something of certain degree of significance when we're looking at a variety of physical conditions. But this is more contextual. When you hear about receptor mediated, think about these LDLs. That's the big player. They're demonstrating these particular proteins. Let's say these are cholesterols that are coming in. They're attaching, and basically they assist the formation of this vesicle. Okay? Okay. That's as much as I can tell you. The opposite, exocytosis. So we're ejecting stuff from the cell, and there's lots of different things can do it, different kinds of signals, membrane voltage. We don't have to deal with that too much. Again, things are encased in membrane. The name that we give this is secretory vesicle. When you spit something out of a cell, the name we give it is secretion, secretion. And you use the term all the time. So whether it's perspiration coming from a sweat gland or oil coming from a sebaceous gland, by definition, they are all secretions. When your pancreas gives out insulin, that is a secretion. And indeed, it's spit out that way. It's not like just being, popped out or spilled out of a cup or anything like that. It involves membrane that surrounds it. And so it's a very, very distinctive way. When we get to, and the example is coming up very shortly, when we get to understanding the neuromuscular junction, how the nerve tells the muscle to contract, it involves the release of a particular chemical that down the road you will come to know is called acetylcholine. Acetylcholine, you don't have to write it down now. We're getting there. You'll be sick of it at the end of the term. Okay, so here's how it works. You have these vesicles, and you don't have to know about these. They're called V snares. V stands for vesicle, and T snares represent the membrane. And so basically what happens, this is what I was talking about as a neurotransmitter, mucus, cellular waste, hormones, pick it. So let's look at the pictures. This vesicle has got the stuff in there. Let's say it's acetylcholine at the neuromuscular transmitter. At the neuromuscular junction, you will come to know about. Here's a vesicle, not just a little membrane sphere. It's got little proteins sticking out of it, like, again, phospholipid bilayers have embedded proteins. This is an embedded protein that's protruding. We call it a snare because you'll see what happens. So on the membrane, it's got a T snare. You don't have to know the names or anything like that. Next step, okay, they fuse. And so it's almost like the proteins engage each other by fusing and changing shape. They pull the membrane toward it. Next step is they get pulled closer together, and a pore develops from the action of those proteins, creating a degree of separation. And then voila, out it goes. It spits out effectively. It incorporates that membrane that was previously pinched off. Now it's gone to another area of the cell and being expelled. That's what I mean by membrane being conserved. And voila, out go whatever. The insulin, the neurotransmitters, who cares? This is the actual microgram, magnified 100,000 times. These are the particles. So why is that significant? Because a lot of times when we see certain kinds of toxins, certain kinds of medicines, they can either make that process occur more or restrict it from occurring. Okay. So before I go back and review resting membrane potential, the example is this. And why I cite it, and I'll undoubtedly mention it again at the neuromuscular junction. Why I talk about that, this chemical makes a muscle contract. There's an agent that we derive from one of the strongest toxins that we know of that we actually use therapeutically, except we use it in extraordinarily small doses to prevent certain muscles from contracting. Anybody want to render a guess? If I said to you it has cosmetic implications, does that help? Who said that? You're a winner. We have a winner here. Botox it is. Congratulations. Botulism toxin is one of the most powerful neurotoxins we know. Neuro meaning it infects the nerve. What it's doing is it's stopping the nerve from spitting out chemicals And those chemicals all say the same thing to the muscle: contract. And if they can't get, if the message can't get through, they don't contract. And so it lasts for about 6 months. And it's used for a lot of things. Originally, more cosmetic, wrinkles and things like that. Now they use it for everything, for people who perspire too much and they're concerned about body odor, migraine headaches. There's a lot of implications, though still the predominant usage is cosmetic and orientation. Voila. So you got it. I'm trying to give you context while you learn this stuff. Okay? That's why you'll be very cognizant of those things. Botulism can be, you don't hear a lot about botulism. I talk about it when we get to, but effectively, the ability to make botulism, the bacteria is everywhere. Everywhere. It's in the carpet in this room right now. So is tetanus. They're here for a party. Resting membrane potential. We saw that animation. Go back and look at it. We call it a voltage. We call it polarized. When one side of the membrane is positive, the other side is negative. All the numbers. We use a minus sign. And this is just potassium, the big player in resting membrane potential. Leakage channels. And when they say down its concentration gradient, again, we always have more potassium inside the cell than outside the cell. And remember what happens when it leaks out. We have those wonderful sodium-potassium pumps bringing back in more potassium to maintain that gradient. That's why they're busy. When the potassium's leaking out, we want to make sure that there's still always more potassium inside the cell. And by the same token, they're spitting out sodium, so there's always more sodium outside the cell. That's those sodium-potassium pumps, why they're so important. And the big thing that makes it negative are the proteins. Proteins have a lot of amino acids. And even though you don't have to be familiar with the chemistry of it, you have, remember, an acid looks like that, organic acid, that C double bond OOH. When it loses the hydrogen, it loses a hydrogen ion. So if there's H plus over there, and it loses this right away, because everything's in solution, this has a negative charge. So organic acids, this is the chemistry behind it. Inherently, when you put those acids in solution, the acid part of it, what's left of that C double bond O, is negatively charged. And that negatively charged, and there's an abundance of proteins inside the cell, and they are so large they can't get out. That's why the interior of the cell is inherently negative. The potassium is offsetting it to a certain degree. Okay, and again, potassium is then pulled by the more negative interior. Okay, and then it's balanced out. Now we got minus 90. Remember, the sodium on the outside, even though there are fewer channels, the sodium positives are going to be more attracted to come into the cell and get us down to minus 70. Voila, go look at that. Chloride does not influence the resting membrane potential because its concentration and gradients are balanced. So even though you have chloride on the outside and chloride on the inside, but they're not impacting it. That's why potassium is a big player. And the fact that the proteins can't get out of the cell because they're just so darn large, that's what they're negative. That's the whole basis of resting membrane. And it's in all the cells. It's nothing unique to a specific cell. And they're showing you all that good stuff here. Effectively, what they're saying here is the positives here, even the occasional sodium, are balanced out. So once you get, here's the positive on the outside, negative on the inside. Once you get into the interior, it's neutral. Same thing here. It just so happens that chloride plays a significant role in maintaining the neutrality out here. So effectively, the intracellular fluid and the extracellular fluid are by their very nature neutral, neither positive nor negative, but right along the membrane, not so. That's the big deal. And you're going to learn about that. Again, when we get into, and we'll touch at the end of the class, there's certain neurotransmitters that basically open what we call chloride channels and allow chloride to go in. And what that does is it lowers the electronegativity even more. We'll get into all of that. That's a very interesting thing. That's the way a particular chemical agent works that students are sometimes familiar with, called alcohol. Chloride gated channels. See what you have to look forward to? And you can go through that if you want. So again, the pump, and again, the 3-to-2 relationship. Sodium comes in, we eject 3 out of it, we bring 2 potassiums back in, and it maintains the steady state. The rate of the pumping out of sodium equals the rate of sodium coming in, etc. Nerve and muscle cells upset this state by intentionally opening additional channels for sodium and potassium. You're going to see this when we really study how a muscle fires and how a nerve fires. This is the foundation. That's the most complex area I teach in AP1. And that's why I'm trying to spend some additional time here. I'm not going to sit here now and ask you about that. It's not coming on a test right away. It'll be fairly straightforward questionings about this until we get into the latter part when we look at muscle physiology and nerve physiology. Then we'll look more into that. There. Here are those cams again. And so the question was asked earlier. These are on the test, but it's more definitional. Again, the cell adhesion desmosomes are the strongest. Tight junctions, think watertight. The more you have of those, the less likely fluids can get by. The connexons are gap junctions for signals. That's the basics that you have to know. Okay. Keratin is involved in the intermediate filaments. It's a structural protein. That's the desmosomes are about that, holding things. Think strong. That's the big deal. But there's other things about it as well. So always involve that outer shell, cell adhesion molecules, the plasma membrane receptors. This is how cells interact with their environment and directly with other cells. So the part of this is kind of showing how one cell influences another. Might be the way to express that. So we have that. And it really is significant because when it's altered, we know there are problems. Okay, every cell has all those glycoproteins. It has adhesion molecules. And these are just the things we talked about, the role of the proteins and things like that. They anchor the cell to the extracellular matrix, think connective tissue, or to each other, think epithelial tissues. Cells slide over one another. If they're not tethered into place, bound into place, they're moving around. Think, say it 1,000 times, think dynamic. We're so used to seeing things two-dimensionally. In our minds, it's static. But you've got to understand it's a living, breathing entity, even in the interior of the cell. Sort of speaking, trying to create a certain degree of imagery. And okay, things like that. Chemicals are released, and white blood cells. It's all about chemical. The white blood cell has no idea you cut yourself. It has no idea you sprained your ankle, stubbed your toe. It does not. Okay, we've got a burn in the kitchen. Okay, chemicals are released from damaged cells. They get into the bloodstream. We call them cytokines. Make cyto for cell, kines for kinetic energy or movement. Okay, so effectively, they're sending these signals in the bloodstream to wherever the white blood cells. It's like they're on Price is Right. Come on down. You can just see that. I love that. Price is Right game show. I remember when it was in its infancy with a guy named Bill Cullen, who was a war veteran who lost his leg. You have to go back historically into the annals of Price is Right. It wasn't nearly as modernized as it is today. And why does the person who's in last, they should win every time. You know, the next time the person goes, oh, 1,200, and they go 1,201. Fine. If it's over, you win. Or they'll say $2 if they think it's lower. But the person will say 1,200 and they go 11.99. I want to throw something at the set. How can you do that? How stupid can you be? It's like missing the puzzle. You know, when they have all the letters out and you can't pronounce it properly. Are you kidding me? Bring back big bucks, no whammies. Nevertheless, stimulate synthesis or degradation of junctions. So there's lots of different things that they do to either make things less tight or more tight, signals. Okay, Typical. Cells that are in contact recognize each other again by the receptors and the carbohydrates that are attached. Okay, normal development and immunity. So very well, again, it's not a visual thing. It's not a sensory thing per se. It's all about touch. In that case, it's sensory. Chemical signal. Binding triggers activation, opens different channels. And that's what we'll look at when we start looking at neurotransmitters or hormones or even local signaling things called paracrines. We'll get there. Right now, paracrines are more of an AP2 type of thing. So that's different kinds of ways that the membrane works. So different responses in different cells. Okay, and to understand that, you'll hear, and I mentioned the name before, acetylcholine. Nearly all the time, acetylcholine is positive. It makes something happen. Very rarely does it cause things not to happen, but there are exceptions. And so you can't, the broad brush thing when you're dealing with anatomy and physiology, there's very few alls and very few nevers. Okay, and so all those types of things, and we are going to learn about whether it's opening or closing gates, whether charged particles are flowing, whether chemicals are flowing, blah, 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 blah. They all have a different role. And the reason I'm presenting this to you, you will be able to see why some things work faster than others. Some signals we hear, biologically speaking, we get the message faster than others. You can see when charged particles are moving around, a lot faster. I'm going to show this to you. I mentioned it in passing. This is the way, for instance, the best contacts, how does insulin? Insulin's too large to get into the cell. Remember, it has a contact called signal transduction. It starts a cascade of reaction. This is the cascade. You do not have to know all the different steps that are in there. G protein receptors. The big G stands for a chemical you're already familiar with. Guanine. You're going to see in that, you're going to see an activated form called GTP. Remember, guanine and adenine are both purines. Because of that, GTP behaves similar to ATP. So chemically, that's the change. So these G protein receptors activate G proteins because they have guanine sort of as a foundation. And you're missing the best part. You'll get over it. Ion channels activate other enzymes, but release of internals they call second messenger. Insulin is the first messenger. It can't get into the cell. But what happens, those changes it makes release a series of messengers inside the cell we call second messengers. And most of the time they're things like calcium, or remember I told you ATP, when we break it down, has multiple actions. This is AMP. That's ATP having lost two of its, those two high energy phosphate bonds. See you later. But AMP, that adenosine monophosphate, with one phosphate, is able to change its shape just a little bit and make like a little circle, hence the name cyclic AMP. So here it is, and it's a relay race. G proteins relay the between extracellular and first messengers at outside guide. They call a ligand, insulin, and create second messengers inside. It's a little easier to show it this way. And again, I don't love the illustration. So the first messenger, called insulin, bonds. Okay, this is this G protein. It's huge. And what happens, it changes shape here and causes a chain reaction. You don't have to go into all the things that they are or anything like that until eventually it goes along the internal part of the membrane, changing shape, triggering one to change, and the next, and finally, it gets to what we call the effector protein, typically an enzyme, and what it does, it activates what we call a second messenger, like calcium or cyclic AMP, and then we have all sorts of activating multiple reactions. You don't have to know these steps, and I typically, when I say that, the ears shut off, but understand that's why insulin has multiple roles. That's why human growth hormone does multiple things, because it activates this cascade of reactions inside the cell. and you can look through this. It's a terrible graphic. The relay race idea and passing on the baton, if that works for you, good. Most of the textbooks use that. That's fine and dandy. Blah, 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 blah. And they activate other things, other chemicals. When they activate chemicals, it takes some time. When they activate ion channels, charged particles moving make things happen fast. You're going to see that, not for now, down the road. I'm so happy. That's probably the hardest part of this early stuff when we look at the cell. The next PowerPoint deals with good old parts of the cell. That's right, nucleus, mitochondria, ribosomes, lysosomes, smooth and rough endoplasmic reticulum, good to their mother. Well, every Mother's Day, all that good stuff. I see the excitement. The enthusiasm is palpable. The Mets should send a thank you note to the Pirates for beating Cincinnati. You baseball fans. As a diehard Yankee fan, you're always looking for the Mets not to win. Baseball is a sport. You've heard bats, balls. I know. We have the Pirates. My wife throws things at Zen. Voila. Parts of the cell, run on one. You've heard all this before. Okay, yes. Composed of this sol gel. Cytosol says solution, water, and different molecules that are in there. Because they're in, they can, and you're going to see this, they can go from being dissolved to forming solids, relatively speaking, protein, salt, sugars. That's why we call this a sol gel. It's not just a uniform liquid. It has a lot of different molecules that aren't. Granules, storage granules for energy, pigments in certain areas. Nerve cells have a fair amount of pigment. A lot of that pigmentation tells you it's making those chemicals I've started to talk about called neurotransmitters. We see pigmented areas. Well, for instance, we'll see a part of the brain where they appear red when you stain, and others where it sends black, both in the same area. That tells you there's neurotransmitters being made, fatty droplets, different kinds of vacuoles for storage of fluid, different kind of crystals. All of these are part of individual cells. I'm not going to go into that. I'm just trying to give you a general feel for it. And what they have a lot of are organelles. They are not all membrane bound. An organelle, again, it doesn't have to be part of this membrane bound organelle system. The ribosome is not. The ribosome, only if it's doing something very specific in the proteins it's making, does it become bound to membrane. You're going to see the difference between what are called free ribosomes that are not. They're making proteins that you're going to use inside the cell. Ones that are going to be used in membrane containers or spat out by that exocytosis or in the cell membrane, they're what we call bound ribosomes. So here we are. The ones that are membrane bound, famously the mitochondria, energy factory. And so inevitably, I haven't met anybody who gives tests that doesn't have some kind of matching with one of these. You'll have like, this will be A, B, C, D, and E, and then there'll be a little thing, and you have to figure out which is which. The energy plant. Hey, it's mitochondria. Okay, wait till you get to the thing called the nucleolus, this little nugget inside the nucleus. It makes ribosomal RNA. That's on every single test I've ever made. Did you hear that, everybody? I'll repeat that. Every single test. Nucleolus, where ribosomal RNA is made. See, anybody who gets that wrong, I become incensed. What does that tell me? Someone didn't, this is going to shock you, didn't study at all. Okay. Mitochondria, energy factory, endoplasmic reticulum. That's all these membrane corridors. Golgi apparatus, packing and shipping department of the cell. Okay. Peroxisomes for defense. But what you're going to learn about are called toxic derivatives of oxygen. That's right. Too much oxygen is bad for you. Lysosomes, we mentioned that already. Digestion and defense. And ribosomes, by definition, are not bound in membranes. The inside of the cell is very well organized. It has fluidity. Things can appear and disappear. Those building areas I was talking about, having an arm that's there and all of a sudden that starts to disappear and now it grows out here. That has those are parts of what we call the cytoskeleton. So we have things that assemble and disassemble. And areas, the centriole is a neat place where one parts of the cytoskeleton goes from. You probably learned a little about that when you learned about elementary mitosis and meiosis, that old spindle thing. Remember that? Just love that. See, it doesn't, for your generation, it doesn't necessarily. My mom was ecstatic when she got this thing called a clothes dryer. It used to be, you'd hang the laundry down in the basement, or if it was nice, you put it on the clothesline. That's kind of what the spindle reminds. I know. Grew up in my early years in the city where you shared the clothesline with the people across the way. I have to contact my sister and see if she remembers that. She's older than I am, thankfully. I tell you, we're having a party on October 9th, even though you're not going to be. We'll have it on the 10th. My sister's going to be 80.
Yes.
What gag gift can I give her? I know. When she was 70, it depends. The worst part about her, and she's great, brilliant, wonderful, she weighs the same as she did in college. Both her and her husband. Freaking the same. I have a picture. It's disgusting. She weighs 97 freaking pounds. I have a leg that weighs 97. Oh, I can't eat another bite of that half a sandwich. I won't fit into my size too. Oh yeah, thanks. Yeah, anytime I get on the scale and it doesn't have a 2 in front of it, I'm celebrating. I digress. Moving on. Mitochondria. Maybe the most important one to talk about. Yes, famously, the power plant. Okay, that's where something called aerobic respiration occurs. Whenever we bring something in, like sugar, basically we start to break it down in the cytoplasm. But in order for it to really be converted to a substantial amount of ATP, or energy currency, it has to have oxygen. Oxygen is like the ticket to get into the mitochondria. It requires it. And the fancy name we give it is cellular respiration, because effectively we're taking in sugar and oxygen. We're creating is the wrong term. We're generating ATP in an energy currency, and we're releasing waste products like carbon dioxide and water. It's a very, very famous reaction. And then really the foundation reaction is what we understand. If you take glucose in the presence of oxygen, we've got some plus oxygen. Go with it. Reversible reaction. And we generate waste products, carbon dioxide and water. And then we get energy from it. The reverse of that, that's typical respiration. The reverse of that, going in this direction, the famous name is called photosynthesis. Now plants, they go out and make a plant. They take energy effectively from the sun, take waste products, carbon dioxide and water, and voila, we get carbohydrates, an energy source for us, and oxygen. You've got to go over and say, well, thanks very much. I appreciate what you're doing for me. Green guys out there, nice to see you. It's like, have you hugged your plant today? I'm going a little over the top of it. So, that's it. Now, surface area. How can this stupid little thing have so much ability? It's this. The inner membrane has folds. Again, biologically, the more reactions, the more surface area. We're taking a lot of membrane, fan folding at that thing again, because it's another membrane. It's like a double membrane. There's a membrane inside of a membrane. We fan fold the interior membrane, and it's embedded with proteins, and effectively we're moving charged particles around. That's how it works. You don't have to go into it. We're not going to talk about the Krebs cycle. You're not going to have to memorize the steps of it or anything along those lines, but that's really important. They call the cristae, these folds, so surface area embedded with membrane proteins. They don't just play a role, they're vital to it. Okay? And this is the unique feature. The mitochondria contain this, the only other organelle that has DNA. You did not know that. That's very important, because understand, over the summer I was going to the gym, 2-3 days a week. My muscles were getting larger or more active. In order to support them, the mitochondria are able to replicate. They have their own DNA. But, school started, I got a little lazy, we got busy, all this type. I'm doing a lot of teaching. All of a sudden, I'm not going as much. The mitochondria, which have a lifespan, don't get regenerated. Ergo, they disappear. Mitochondria do that. If you're using your nervous system, if somebody unfortunately has a stroke or something, the mitochondria and wherever the target is are going to start to disappear because they're not getting any nervous stimulation. That's the way it works. The DNA, you'll come to know, comes from a very interesting source. We talk about it in micro, called endosymbiotic theory. It's interesting because it's very, very similar. It's not like the double-stranded DNA, that alpha double helix in the nucleus. It's a circle of DNA. That's very much like bacterial DNA. It's a very famous theory called endosymbiotic theory. You don't have to know it now. That's micro. And the same thing with the RNA is very much like bacterial RNA. Okay? So they have their own DNA and RNA, so they're almost like mini cells inside. They're really not true cells because they can't exist on their own, but they're mini cells, if you want to think of it that way, that have the ability at least to make more when needed, or to stop making more, and effectively the lifespan will disappear. They resemble bacteria. They can divide. The division is called fission. Bacteria don't do mitosis and meiosis. They do fission. They just keep dividing in two. Got it? So that's a foundation for whoever unfortunately has you for micro. We'll have to tell you that. It may be me. I need a drink on that. So you wonder what's in here. That would get me fired. Voila. This is the microgram. Look at all those folds. So it's got an outer membrane that's permeable. All membranes are permeable. And it's got an inner membrane. Okay, and that inner membrane, fancy name, we call it the crystal, all of these folds. And basically all we're doing is we're pushing protons around, generating gradients, and allowing them to come back through and spin yet another enzyme called ATP synthase, fancy name. And it's like a turbine. It's like if you go to Niagara Falls and the water's coming over the fall, hydroelectric power. The water is going through and it's spinning it like that. The alternator in your car is doing that. The belt's spinning and causing something to move and keeping your battery charged. But you were a dummy like me about 2 weeks ago and left my lights on. My boss had to jump it out here in the parking lot. So here, the enzymes are the chemicals that are stuck in there. It's a very accurate representation. The mitochondrial DNA is circular. You don't have to know that from me now. And the ribosomes are interesting in their construction. Moving on to the ribosomes. Okay, two types. I touched on it before briefly. They are not by definition membranous organelles. They only become one when they become membrane bound, when they attach. And that's when all they're doing is making proteins for a specific purpose. They attach to the endoplasmic reticulum, which we'll look at. And these proteins, by definition, either have to be incorporated in membrane or bound in membrane or spit out of the cell to do other things, exported out of the cell. So you're going to see that, and we'll go over the three destinations that are there, and they're switchable. The DNA that comes out has a little signaling molecule when it attaches to the ribosome, the messenger RNA, when it comes out of the nucleus, when it attaches to the ribosome, will tell it where to go. If you're a free ribosome, just make proteins here, or we need you to go over and become a bound ribosome. Okay. And switchable is a very way to do it. The ribosomes are made of ribosomal RNA, that's right, from the nucleolus, and protein. And you'll take a look at that in a second. We're not going to do that now. I'll stop it here in the interest of time. And then we'll start with the endoplasmic reticulum. It's really the best way to start to discuss something that you will have to know called the endomembrane system. Elaborate membrane passageways that start at the nuclear membrane. Thank you. So Friday, I will be here. Not next Friday. Mark it down on your calendars.